All right, in section 8.2, we're going to be talking about the structure of DNA, how it's built, and how did we come to know about its structure. And what we know, number one, is that DNA is, as far as structure goes, is the same in all organisms. All organisms have the same basic components and structure. And this video we'll take a look at tomorrow, so we can talk about that tomorrow in class. But to start with, DNA is made up of 4 nucleotides. Nucleotides are the basic building blocks. So remember us talking about polymers. Nucleic acids are polymers, just like lipids are polymers, proteins are polymers, carbohydrates are, nucleic acids are too. And if you remember, I said we're not going to talk about DNA and RNA right now because we're going to spend a whole chapter on it. So this is the chapter where we're going to be talking about DNA. So if DNA, which stands for deoxyribonucleic acid. If that nucleic acid is a polymer, the monomers of it are nucleotides. And so DNA itself is what we call a double helix, which is 2 long chains of nucleotides that are twisted in kind of a twisted ladder shape. The nucleotide, or the monomer, as you see right here, of DNA is composed of 3 smaller parts. So this one thing right here by itself is a single nucleotide, but it's composed of a phosphate group, a 5-carbon sugar, and in DNA it's called deoxyribose. Right ends in ose, so it's a sugar. And then it also has a base, a nitrogen-containing base. Now, as far as the bases go, they are the only thing that's different in our DNA molecule. Everything else about DNA is the same. The sugar phosphates and are the backbone. If you're talking about it like it's a ladder, the sugar phosphate components of the nucleotides are the backbone of the ladder, and these nucleotides make up the rungs of the ladder. And so you can see that they are grouped into 2 different categories. The pyrimidines, which are thymine and cytosine, or your T's and your C's, are a single ring structure, and your purines, which are adenine and guanine, are double ring structures. So how did we come to find out what the structure of DNA was? Well, there was lots of different tests going on and studies going on, but the credit for the 3-dimensional structure of DNA, or the 3-dimensional model of the structure of DNA, was discovered by Watson and Crick. And they were doing lots of different setups. They're actually building this in a garage office out of cardboard and wire, so it wasn't a very fancy model to begin with, but they were doing all sorts of things. A 3-strand structure, a structure where the nucleotides were on the outside of the molecule instead of the inside. But eventually they came to realize that the double helix has a backbone of sugar phosphate on the outside. So again, if it's a ladder, the sides of the ladder are made up of a repeating sugar phosphate chain, if you will, and the inside, or the rungs of the ladder, are made up of the bases. Now, what they discovered was actually built off of the work of Rosalind Franklin and Erwin Chargaff. These 2 worked together. I wouldn't call them close working friends, but they were colleagues, Franklin and Chargaff. And Franklin was known for her abilities with X-ray crystallography, which basically meant that she would remove the water molecules out of a subject, or out of a substance, and take an X-ray of it and look at it. And so she got a very good image, and that's the one you see right down here. That's photo 51. That's what they call it. Probably because it was the 51st picture of DNA that she tried to take. But that X-ray image told her some stuff about DNA that Watson and Crick didn't know. One thing was that the double helix was an even width all the way across. And along with finding out, so the story is that Watson and Crick came upon Franklin's X-ray image not with her approval. She didn't know they saw it. They were given a copy or shown a copy of it. And when they were shown that copy, that's when they figured out what they were doing wrong with their model, and figured out the even width, as well as remembering Chargaff's rules. Now, Erwin Chargaff was an American biologist who studied DNA from all sorts of specimens. And although he found that not one specimen had the same amount of DNA, what he found was that in all specimens, whether you're a plant or an animal or a fungus or whatever you are, your amount of adenine always equaled the amount of thymine. So if he took a DNA sample from your hair and ran it, however much cytosine he found in that sample, there would be just about equal amounts of guanine, and the same for adenine and thymine. And that was the same in all species. So knowing that, that's how Watson and Crick built their model. So these base pairing rules that Chargaff came up with show how nucleotides always pair. And so A always pairs with T and C always pairs with G. And that is how we get that even width across, because you have a double ring structure. Remember, one is a purine, double ring. One is a pyrimidine, single ring. And so you always get that even width across, because you always have basically then 3 rings going across. 2 from your purines and 1 from your pyrimidine. So you always have a nice, even, uniform width. When you look at a model of DNA, okay, you see that we have the structure here. The backbone would be the blue part. So you have a sugar, phosphate, sugar, phosphate, sugar, phosphate, right? Backbone. So this guy right here, this phosphate, this sugar, and this base, right, our T right here in the beginning, this is one nucleotide. So this side of DNA is composed of 4 nucleotides. This little segment of DNA is composed of either 8 nucleotides or 4 nucleotide pairs, 4 base pairs. So the nucleotide is connected to the next nucleotide by this bond right here, and that is a covalent bond. Covalent bonds, remember, are nice and strong, and so they're holding that together. The hydrogen bonds then are the bonds that hold our bases together. They're much weaker, but when there's so many of them, they do a good job of holding the molecule together.